Hello and welcome everybody. In this short video, we want to talk about GRINopathies. What are they, who is affected by them, and why a personalized therapy is needed. First of all, we want to introduce ourselves. We are two research teams led by Dr. Xavier Altafaj and Dr. David Soto, two young researchers who work at University of Barcelona and at the Research Institute IDIBEL. With this video, we want to inform you about our research project and explain why we need your support. Our principal goal is to carry out a study which allows us to develop personalized therapies for GRINopathy patients. But what are GRINopathies? To understand this better, we need to provide you with some basic concepts about neuroscience. Will you join us? First of all, we'll explain what genes and proteins are. In a simple way, genes are DNA fragments whose goal is to encode instructions for protein synthesis. They're like a handbook. Each gene is an instruction, and every instruction will provide us with a protein. Proteins are essentially the functional engines of the cell. They have functions such as physical transport, chemical reactions, catalysis, or they provide a supportive function. One of the functions of these proteins is to receive signals from outside of the cell. Receptors are the kind of proteins that play this role of receiving messages. Each receptor has to answer to a concrete message. In other words, they act in response to a particular substance. Depending on which kind of receptor is activated, the signaling inside the cell will vary. As in the rest of our organism, our brain contains a great variety of cellular types. Amongst this variety, neurons are the ones involved in the information integration and transmission. They're essential for learning, motor coordination, and more complex aspects like memory storage. Well, what kind of role are receptors playing in neurons and in the nervous system? Receptors play a fundamental role in the neuron-to-neuron communication. Due to this function, receptors allow neurons to work correctly. Communication between neurons is placed in a highly specialized structure called the synapse. In this process of neuronal communication, neurons send messages from a presynaptic neuron, and the postsynaptic neuron contains the receptors to receive the message. There are many kinds of synapses, neurotransmitters, and receptors. One of the most important receptors are the NMDA type receptors. When they don't work properly, pathologies arise like the GRINopathies, which is the main topic of our research. As previously mentioned, genes are the handbook to create proteins. GRIN genes are the ones who have the information to create the NMDA receptors. In normal conditions, GRIN genes can provide the information for NMDA type receptors, and these receptors can exert their function properly, such as memory storage and learning processes. However, when the DNA contains mistakes, we refer to them as mutations. These mutations can be responsible for an incorrect function of the NMDA type receptor. If the receptor is not working correctly, neuron-to-neuron communication is not correct, and this is translated into a nervous system disorder. Mutations at GRIN genes affects the cerebral development from the very first life stage, and their consequences last a lifetime. At the clinical level, the effects of the mutations in the GRIN genes show as intellectual disability, motor dysfunction, and in some cases in concrete types of epilepsy or autism-like spectrum. Today, no treatments exist to repair NMDA receptor damage. However, it has been shown that with pharmacological treatment, these receptors can partly recover their function, which allows to improve the patient's lifestyle. Unfortunately, there are many mutations that cause GRINopathies, and not all affect the NMDA receptors in the same way. For this reason, it is necessary to perform functional studies of each mutation to give the children who are affected a better and personalized therapy. Our groups have the equipment, the knowledge, and the passion. However, the main problem at this moment is the lack of funding to hire qualified staff to perform the great amount of experiments that this project will need. Time is ticking and we need your support.